Here we go with our second type of macromolecules part D, and lipids. So lipids are a very diverse group of hydrophobic molecules. And basically, there are three main types of lipids, and what they have in common is that they're hydrophobic. And so we just kind of put them all into this one category because they were all hydrophobic. Okay. Lipids also do not form polymers. Okay. So the unifying feature, as I just said, of all lipids is they have little or no affinity for water. They're very hydrophobic. And they're hydrophobic because they consist mostly of hydrocarbons. And the hydrocarbons, as we know, are nonpolar. We're going to look at the most biologically important lipids, which are fats, phospholipids, 2 and 3 steroids. So let's first talk about fats. Fats are what makes your food taste good. So fats are made up of two types of smaller molecules, glycerol and fatty acids. So glycerol is a 3-carbon alcohol with hydroxyl group on each carbon. And a fatty acid is basically a carboxyl group with a very long carbon skeleton. And you'll see they make these long, long chains. And there they are. So here is my glycerol. Okay. And you'll see it has 1, 2, 3 spots where a fatty acid, here's my fatty acid, can be brought in to join up with the glycerol group. Okay, so this is one of 3 dehydration reactions and the synthesis of a fat. Eventually, there will be 2 more fatty acid chains here, and we'll have 3. Fats separate from water because water needs to form hydrogen bonds with polar molecules, and fats are non-polar. So they're hydrophobic. So when we get our 3 fatty acids joined up to the glycerol, it creates what we call a tri-3 acyl glycerol or a triglyceride. And I know you guys have heard of these. Okay, so it's part of like your blood work or dietary, you look at your triglycerides. Okay, so here is a triglyceride, and you'll see there's 1, 2, 3 fatty acid chains hooked up to my glycerol molecule. And again, these long hydrocarbon chains make this molecule extremely nonpolar. So fatty acid, the long chains vary in length, or number of carbons in the skeleton, and in the number and locations of double bonds. And the double bonds, the presence or absence of double bonds, depends whether or not, or dictates whether or not, rather, that the fatty acids are saturated or unsaturated. And also, probably you guys have heard of saturated fats, unsaturated fats, maybe didn't know what they were talking about. And what they're talking about is the presence or absence of double bonds. Saturated fats have no double bonds. And what they are saturated with is hydrogen. So saturated fatty acids have the maximum number of hydrogen, so they have no double bonds. Unsaturated fatty acids don't have the maximum number of hydrogen, so they have 1 or more double bonds. So yes, double bonds, bonds, and no double bonds in saturated. So our saturated fats right here, if you look at the hydrocarbon chain, you'll see there's no double bonds present. And because of that, the hydrocarbons chains or the fatty acid chains get very straight. And here's a ball and stick model of looking how straight it is. Because they're straight, you can stack the fats close to one another. Okay, so the molecules of fat can get very close to one another. And when molecules are close and still, you have a solid from chemistry. So saturated fats tend to be solid. So you've got butter, lard, Okay, and this is how we know saturated fats, while delicious, the saturated fats is what makes your food taste amazing. Butter, oh my God. But butter, lard, they're all not good for you because they are saturated. Okay, you get more fat per, say I have, a gram of butter versus a gram of an unsaturated fat like an oil. We'll get to that in a minute. Because it's a solid versus a liquid, there are more molecules in the saturated fat. So I get more fat for the same amount of stuff. Okay, so, and here's our unsaturated fats. If you look in the fatty acid chain, it doesn't need to be all of them. It can just be one. There's a double bond. Voila. And this double bond, we hope, in our unsaturated fatty acid is a cis bond. And cis fats are good. All unsaturated fats are better than saturated fats, but cis unsaturated fats are better for you. Okay, because of this double bond, okay, the fatty acid chain gets a kink in it. So it's slightly bent. So the molecules of fats, the unsaturated fats, they can't stack up next to each other. So because the molecules can't get close, these fats tend to be liquids at room temperature. Okay, so olive oil, coconut oil, all that kind of good stuff. So better for you than butter because it's a liquid and it contains unsaturated fats. Again, fats made from saturated fatty acids are called saturated fats. They're solid at room temperature because the molecules can stack closely. Animal fats are saturated. These are the bad fats. They taste good, but they're not good for you. Fats made from unsaturated fatty acids are called unsaturated fats. And these guys are liquid at room temperature. And these are your plant fats and your fish fats, right? So you talk about like salmon and tuna and all that kind of stuff. Not so good for the mercury, but a better fat for you than like beef or pork. Diet rich in saturated fatty acids may contribute to cardiovascular disease through plaque deposits. So again, not good for you. Tastes good, but not good. Hydrogenation, okay, converts unsaturated fats to saturated fats by adding hydrogen, okay? And by hydrogenating vegetable oils, you can get things like fats, unsaturated fats with trans double bonds. And you guys have heard of trans fats, okay? Trans fats look more similar to a saturated fat, and so they are actually almost worse for you than a saturated fat. So they are super sad. Wah. Major function of fats is energy storage. Okay, so they can be, when we start to talk about glycolysis, they can be converted into ATP through similar processes. I'll refer back to that when we get to that chapter. I think it's chapter 6, maybe 7. I don't know. Doesn't matter. And humans and other mammals store their fat in adipose cells. And adipose cells, like that is what cushions vital organs and insulates the body. So you need fat to help keep you warm and padded. But you don't want excess fat. Our second type of lipid, so that we're done with fats, our second type of lipid are phospholipids. And phospholipids are super fancy. They look very similar to fats, the triacylglycerol or triglycerides, but they only have 2 fatty acid chains. And the third spot on the glycerol has a phosphate group, okay? So actually, let's take a look at it. Here we go. So our 2 fatty acid chains here, there's one, and you'll see that one's saturated, but this one is unsaturated, so it has a kink. So this area is hydrophobic, but that third location on the glycerol is attached to a phosphate group. And holy Jehoshaphat, look at all those electronegative elements. So this area of the phosphate group is extremely polar. So it is hydrophilic. So you have this molecule that kind of has this dual nature. It has a hydrophilic head and hydrophobic tails. And you're going to see phospholipids kind of drawn like this little ball with two tails a lot. Okay. Molecules that have two natures or two types of areas are called amphipathic. Okay. That's a good word. Use it. All right. So when you put phospholipids in water, they have part of them that likes water, right? The head is hydrophilic, and part of them that doesn't like water. The tails are hydrophobic. So when you put them into water, they'll actually self-assemble into a bilayer. The tails point inward away from the water, and the heads point outward towards the water. Okay? And phospholipids is the major component of our biological membranes. Okay? And that is huge. And that basically, when we start talking about the evolution of cells and the evolution of life, phospholipids played a major, major role in that. Without phospholipids, we do not exist. So here's a little bit of look at how they look. So we have our hydrophilic heads facing the water, the hydrophobic tails hiding here inside of it. And what it does is it creates this barrier, okay, that helps separate, say, the outside of a cell from the inside of a cell. Our last type of lipid are steroids. Steroids are characterized by carbon skeletons that have 4 fused rings. And a couple slideshows back, a couple screencasts back, we talked about carbon and functional groups. And I think I showed you a picture of estrogen versus progesterone, both steroids, as we know. They had those rings and they just differed in their functional groups. And that's kind of how it is with all steroids. So today we're going to look at cholesterol, as you may have heard about cholesterol. It's a component of animal cell membranes. Okay. Not high amounts of it can contribute to cardiovascular disease, so it's not so good. But here is what a steroid looked like. So these 4 rings here are basically a common part of every steroid. You see these interconnected rings, you think steroid. You think fat, not fat, you think steroid, you think lipid, you think hydrophobic. Again, it's all hydrocarbon. And then different steroids have different functional groups. So this one has a hydroxyl group, a methyl group, here's another methyl group, and then there's another hydrocarbon chain over here. Okay, and this again just adds this molecule, this little oxygen here on the side cannot beat how nonpolar this molecule is. Okay, and that's it for fats or lipids. The end.